Our next, Sara Monlinari, is a postdoctoral researcher at Rice University in Carolyn Aho Franklin's lab. She completed her PhD with Professor Matthew Bennett, graduating with a thesis on engineered cellular differentiation in bacteria. That work was part of a DARPA effort to develop living materials. And that helped her develop a great interest in the field, and therefore she joined Carolyn Aho Franklin's lab, where she's now working on engineering artificial biofilms. So thank you for joining us, Sara. Go ahead and unmute yourself.
I have to unmute first and then reshare. Not sure why. So, can you hear me now?
Yep, looks good, sounds good.
Thank you so much for the opportunity to give a talk to this meeting, and thank everybody for dialing in. Today I'm talking about what I've done during my PhD thesis, which is like a synthetic system for E. coli to divide asymmetrically. She was explaining before, this is like part of a larger effort to try into engineering living materials. And the reason why we want to do that is because if we look at actual living material that exists in nature, we can see they have remarkable characteristics, such as self-assemble or respond to stimuli. If you look at a plant, the plant is spontaneously able to grow toward the sun. Or the liver, for example, which is able to self-repair upon an injury. The skin, same thing. So these are like characteristics that are not present right now in artificial materials. So the goal here is that can we mimic some of the characteristics of the living material so we can adapt them and reproduce this extraordinary ability to man-made material. One of the things that these all materials have in common, despite being so different, is the multicellularity. So the fact they're made of not just one clone of cell, but different clones of cell that works together to create the final properties. If you ever look into a piece of wood, for example, you can see there's a lot of different cells that have a different morphology. And in this way, they can be more robust because they can distribute biochemical and biophysical tasks. They can also coordinate to each other. Some of the cells will lose completely the reproduction potential, for example, and differentiate to have highly specialized functions. So it's a little bit the concept of divide and conquer. So what we want to do here is that having a bacterial population, which traditionally would divide and provide identical clone, that is able to differentiate and create different cell lines that potentially can be engineered to do coordinated function. Ideally, like if you look at a material, there's not only just 2 or 3 different cellular types, there's like an unlimited number, like very high number. So we don't want to just differentiate them, or we're trying to expand really the amount of clones that we can get. We want a system that is like a platform to be further engineered into a more advanced and highly capable differentiation system. This is not like a new attempt. Like people before have tried to differentiate bacterial population, and one of the most remarkable example, which also gave rise to the whole synthetic biology field, was the toggle switch. But for the specific purpose, the toggle switch is a little bit weak because it's very subjective to noise, extrinsic and intrinsic noise. So a single cell flips continuously from a state to another. And you can imagine that if you want a cell to be differentiated to carry on a function, you want that specific cell to hold the transcriptional profile as long as you can. So if the single cell, for example, has to travel in space, it will not be able to do so if it loses and regains motility and flips back and forth all the time. So what we really want to do is a more stable system that resemble what happened in eukaryotic systems, where the cells divides and create two different cells at the same time. One cell, it's identical to the progenitor cells, while the other cell differentiates. And the differentiation is ideally terminal, like final. In particular, if you examine from an engineering point of view, we want a system that is able at the same time to do symmetric and asymmetric cell division. Because what we really want to do is want to be able to control the onset of asymmetric cell division. And as I was saying before, we wanted the differentiated cell to be terminally differentiating, meaning that the differentiated cells could never go back to the progenitor cell. Maybe like all of you are familiar with the concept of plasmids in bacteria. There's many different bacteria that can grow in E. coli. Most of bacteria have plasmids, and what happens is that in order for the information to be preserved, bacteria make sure that when the cell divides, every daughter gets at least one copy of the plasmid. The way we trying to engineering asymmetric cell division was actually through asymmetric partitioning of the plasmid, which means we want to make a system that takes all the plasmid in a unique cluster. And so when the cell divides, as you can see here, it goes necessarily to one side and not to the other, thus creating different cells, different daughter cells. And the red, like the pink colored daughter cells here is actually identical to the progenitor cell because it has the genetic information on the target plasmid, while the gray cells lost permanently the plasmid and it became in this way terminally differentiated. The way we engineer this system is through a protein called PARB that was slightly modified with a leader peptide. This protein has a DNA binding ability that is mediated by a leucine zipper domain, and it has the ability of recognizing a specific sequence called PAR-S and tightly bind to it, but also it can, through a system of interaction, DNA protein interaction, can form a huge nuclear protein complex around this PAR-S. And it's exactly this protein-protein interaction that we want to use to bring plasmids together. So what we've done is we clone this PAR-S on a plasmid, and when we start the induction of PAR-B, as I told you before, PAR-B will first bind to PAR-S, so fill up the region around the PAR-S sequence on the plasmid, but then through the protein-protein interaction, is also going to bring the plasmid together and make them stick to a unique cluster. At this point, the plasmid are all trapped, and so when the cell divides, of course, this cluster goes just to one side, like I predicted before. So when we first observed it through microscopy, and we set up an experiment where some of the population did not have the inducer, so they have no PAR-B, and in this case, the plasmid that we call target plasmid, it divides and partitions as any other plasmid in E. coli, but upon induction of PAR-B, then you have the onset of asymmetrical cell division, and so the appearance of differentiated cell. When we observed that through microscopy, we could see that going on actually very clearly. And if you look at time 18 minutes, when you you first have the appearance of the first yellow dots, this is the cell that we identify as the progenitor cell, the one that will retain the target plasmid because it has the cluster of the plasmid. And the other cells we assume has lost forever that information, and in fact we see the red fluorescent that is encoded on the target plasmid to fade away in the course of time. So if you follow the daughter cells of the the differentiated cell, you see that they go black and they never reacquire fluorescence again. So after this first encouraging results, we want to actually to put a number behind this effect. So we want to know how many differentiated cells are we actually able to get compared to the initial number of progenitor cells. So we set up an experiment where we screen the cells based on their ability to grow on chloramphenicol. And because chloramphenicol is the resistance marker that is present on the target plasmid. So when the cell loses the target plasmid, this white differentiated cells, it also loses the capability to glow on chloramphenicol. So when we induce the differentiation, we expect a much lower number of cells to be able to grow with a marker selection. And in fact, if you look at this curve, when the blue and black curve on top are the negative control, so the population does not have part B, there's no induction of differentiation, so every cell is able to grow indistinguishably with or without chloramphenicol. When we induce the cell in asymmetric cell division, the green curve, but we do not screen from the presence of chloramphenicol, then all the cells grow. But when we observe the cells that are actually able to grow on chloramphenicol, we see that this is like a much shorter fraction, and the number stays the same over time, which is exactly what we expected. After that, we wanted to know how can we modulate and control this effect. So are we able to actually change this like red curve here? So the amount of the ratio between progenitor and differentiated cells at any given time. And it turns out that was actually very easy to do by changing the amount of inducer in the media. In fact, 0.2 is the maximal induction, is the one that we induced for this previous experiment. And then we went back and used like less and less concentration of arabinose. And we see that the less induction we have of asymmetric cell division, the less differentiated cells we get at the end. And so this is absolutely consistent with the prediction. And there's almost like a linear correlation between the amount of inducer and the amount of differentiated cells. In this, after these results, we want to actually to see how robust is the system on the specific kind of plasmid that we use as target plasmid. What we're using till now was a medium copy number plasmid that has a passive mechanism of differentiation, meaning that when the cell divides, the plasmid would just like spontaneously diffuse to one donor cell or the other. But what happens if there's a high copy number, like 500 copies, will our system be able to hold all of them? Or what happens when there's actually an active mechanism of partitioning that our system has to compete against? And we proved that actually our system is incredibly robust to copy number and also to active mechanism of partitioning. And this is a good thing because it can be used as a module, and synthetic biology is very important to be able to use different kind of plasmid, different kind of bricks for a certain system. We use the system to engineer new kind of pattern formation. So we engineer the cell that they become motile when they loses the target plasmid, and they also turn off the red fluorescence and acquire yellow fluorescence. So if you look at the bottom row, when we induce the differentiation of cells, we see that we have formation of differentiated cells that swim away from the initial core of progenitor cells. In the top row, this is the negative control. All the population is red, meaning they're all made of progenitor cells. There's no differentiation. We also, like I was telling you before, because we just didn't want two different types of cell, but we wanted more, we found a way to characterize an orthogonal system, and then we put both of system in single cell. And in this way, we obtain three different kind of differentiated cells, two of which are partially differentiated and one is terminally differentiated. And we prove that with microscopy and also with the plating assay. I have to be a little bit quick on this part, even if it was extremely complicated to screen all the possibility. And in summary, we created a system for asymmetric cell division of E. coli that is tunable and robust to copy number and origin of replication. It can be used for new kind of pattern formation, which is like the most important feature for us now in terms of engineering of living material. Sorry. And we also identified orthogonal system that can be used to expand the number of differentiated cells that we can get out of a single progenitor cell. And with that, I want to thank all my previous lab, the Bennett lab, and thank you for your attention.